Okay, we are now live on Facebook. So let me start. My name is Daryl Boness. I'm editor-in-chief of the journal Marine Mammal Science. And this is the next presentation in our series for the Society for Marine Mammalogy of Editors Select Talks. These presentations are taken directly from papers that are published in Marine Mammal Science. Just a couple of housekeeping items. If you have questions you'd like to ask of the speaker, if you would put them in the Q&A, not in the chat, although we will monitor the chat in case somebody forgets or doesn't hear this. But please put them in the Q&A. We'll hold all questions until the end of the talk. This will be recorded and posted subsequently on the Society for Marine Mammalogy website. So today our speaker is Dr. Trevor McIntyre, who began working with marine mammals as a field assistant on Marion Island back in 2005, a while ago. He subsequently conducted his PhD work at the University of Pretoria, working on southern elephant seals. After completing his PhD, he did postdoctoral work at the Alfred Wegener Institute in Germany for several years, and then served as a lecturer at the University of KwaZulu-Natal. In 2019, he took a position at the University of South Africa, where he is now as an associate professor. His research over the years has focused on behavioral adaptations to environmental change, particularly with pinnipeds and otters, and he's currently conducting studies on African clawless otters in South Africa and Ross seals in Antarctica. Today, however, he'll be talking about the behavior of recently weaned southern elephant seal pups. And like all phosid species, southern elephant seal pups are abruptly abandoned by their mothers at the end of lactation and are on their own to develop and learn the skills needed to survive. So Trevor will tell us what he and his colleagues have learned about the foraging behavior of weaned pups on their first long foraging trip post-weaning and compare it to foraging behavior subsequently during their first year of life. The paper is based, the talk is based on the paper Tracking the Foraging Migrations of Marion Island Elephant Seals During the First Year of Life, published in Marine Mammal Science. I think probably somebody will be putting up a link to that. The paper is open access, so everyone has free availability to that. On that note, let me turn it over to Trevor to tell us what he's going to tell us.
Thank you. Thanks so much, Daryl, for the introduction, and thanks for the invite to give the presentation. Also want to just thank the student members at large, Teresa and Sophia specifically, for your assistance in putting it all together and making it happen. So let's get going. I'd like to just start off also by just acknowledging all the co-authors who've been involved with this work. As Daryl mentioned, I've been involved with the pinniped program, the Marion Island Marine Mammal Program, since 2005. And that is, in fact, when the first of the animals that we report on in this paper and in this presentation were instrumented. So the data set itself is actually quite old. It spans 2005 through to 2008. And there were various reasons why we took quite a while to sort of get this published. Part of it was me moving between institutions and not finding time to finish things up, and other colleagues having similar situations and holdups. But we're happy to finally have this published now, and I hope you find it interesting. So I'm first just going to give you a quick overview of the presentation. The overall sequence of things I'll discuss would firstly be just providing you with a bit of a background around the ontogeny of behaviors and influences. on southern elephant seals specifically, and then specifically also the Marion Island elephant seals. Daryl mentioned, he sort of mentioned already that they are capital breeders where pups are weaning very abruptly, and that plays a key role in why we're interested in understanding how they're actually learning to forage successfully and avoid predation and things like that. From there, we'll go on very briefly to the methods that we employed with the study and some of the expected results based on tracking data we have for other animals and based on results from other areas within the Southern Ocean as well, where elephant seals have been tracked in the past. The results will largely just be a breakdown of what we found in terms of track durations of animals during the first year of life, the distances that they travel, the directions that they travel in. We'll summarize all of that and then add some potential explanations and questions for further study. So let's get into it. Okay, so in terms of ontogeny of behaviors, the whole rapid acquisition of the ability to forage successfully and avoid predation is really critical for animals, for many of the longer-lived animals during their first year of life. So this concept of, let me see if I can get my pointer working. So this idea of the development of a successful foraging strategy or successful predation avoidance strategy is really important in terms of that first year of life and how animals sort of then survive in order to recruit later on into any breeding population. Now, there are a number of variables and factors that likely influence this, and I've tried to sort of summarize these in a short diagram. These are not exclusive from one another, and potentially all play a role in some populations or some species, but can largely be thought of as either sort of innate qualities or instincts. And this relates particularly to how animals respond to environmental cues. So potentially there's some sort of underlying instinct associated with a species or a population, whereby there is some sort of selective pressure that would allow animals or that would select for animals that show a certain type of response to specific environmental cues. Other aspects that play a role in the sort of development of strategies would include learning from conspecifics, whether that be adults in the same population or potentially even peers in terms of the same age cohorts. Those are potential things that influence this. Intrinsic factors such as the sex of individuals and the condition potentially play large roles, particularly in sexually dimorphic animals, where males may have a very different body size to females and potentially also use their environment in a different way. And then lastly, but again, not exclusively, is this whole idea of learning from experience, where, as you can see from the title of my talk, which is pretty much where this presentation is headed, I suspect that this is maybe what's happening with some of the elephant seals, but there is a question mark over that. Okay, so the fossids, the true seals, are, as we mentioned, they're capital breeders, at least most of them are, which means that this sort of transition to independence is very abrupt. Now, this idea or the fact that this transition is abrupt means that we are taking out one of the variables that would potentially explain how animals learn their foraging behavior. So the fact that they don't have an opportunity to learn from older seals, that pups are born somewhere on a substrate, whether it be ice in some instances or an island or even a continent. The females then abandon them and pups have to sort of find their own way, seemingly completely independently. And this makes them really good study animals if we're interested in trying to understand which factors influence this sort of development of foraging and movement strategies in animals. And as a result, there have been a number of studies on fossids. I've just sort of highlighted a few here. Many of them have been on grey seals in the UK, as well as harbor seals, also in Europe. Those are sort of just some of the studies that I've highlighted on the slide here. So, of course, elephant seals are no different. They're also capital breeders. What's interesting about their sort of breeding sequence is that females would come ashore and give birth, and then there's a sort of short period of lactation before the pups wean. The females would then depart back at sea while the pups remain on land during the sort of post-weaning fast period. And then after a while, they would then go on their maiden foraging trips, which would then take over most of the rest of the year, which would be anywhere from January through to October before they return again for the molt. Now, what's of course very important here is that during this sort of lactation period, so pups are normally born at masses of around 40 odd kilograms as individuals. And then within 23 days of lactation, they basically balloon into these sausages of 120 odd kilograms. And so there's this very rapid acquisition of resources from the nursing females, and then they are left alone. They're simply just left, they hang around waiting for some sort of cue to leave the island and then go and forage. This is most likely just associated with loss of condition, and then that would eventually lead them to going into the water. What is also important is, particularly on Marion Island, we'll get to a little bit of details around Marion Island, where it is, those sorts of details in a second. But what tends to happen is the pups leave the island here towards around January in the year, and then they would go on these initial foraging trips, but then also return back to the island and do stopovers back and do a second or a third or sometimes even a fourth foraging trip within that sort of first year of life before they actually return for the sort of molt haul out. This is very different to the typical breeders in a population. So adult breeders would normally only haul out twice a year. They'll be there once for the breeding season around September, and then once for the molt, which is depending on the sexes and the age groups, would be from December through January for the older animals. So what you'll see in what we're reporting here are sort of these initial maiden foraging trips that were all completed before they returned for the molt. Now, just a bit of background on elephant seals in terms of their distribution. Of course, they have a circumpolar distribution, meaning that they have or that they occur on subantarctic islands right around Antarctica, with the biggest populations being centered around South Georgia, the Kerguelen Islands, as well as Macquarie Island. Now, Marion Island sits over here. It's a very small population comparatively, and this is where we are focusing our study. The big benefit of the Marion Island population is because it is reasonably small, we are able to monitor the population very intensively. So the elephant seal population on the island are subject to a continuous mark resighting program, whereby every pup that is born on the island is double tagged with 2 flipper tags at weaning, and the beaches where they haul out are checked and monitored for their presence at least once every 10 days throughout winter and once a week during summer months when the population is at its peak. So there's this really intensive sort of ongoing monitoring effort. We know individuals, we know their histories, we know when they have been on the island, we know which ones of them have been successful breeders. And that adds a lot of sort of background to why we're really interested in what these under-yearlings, these recently weaned pups, were doing in the first migrations. So, of course, under-yearlings or at least recently weaned pups, their foraging migrations have been studied for other populations and have been described in other populations. What I'm showing here are just two maps. One is for King George Island on the Antarctic Peninsula, where these tracks, these little lines that you see over here, are tracks of also just recently weaned pups. And what you see on the right-hand side is the Kerguelen population, and these individual lines show individual tracks by different individuals. So what you'll immediately notice from these tracks is that while they're not sitting right on top of one another, there's a general consistency in terms of foraging direction. So the Kerguelen animals, most of them seem to forage to the west of the peninsula, perhaps out of sort of denser sea ice, but all sort of in a similar direction. As far as Kerguelen is concerned, the majority seem to travel in the sort of southeasterly direction, with one or two individuals heading off in a westerly direction, doing slightly different, maybe a different strategy, if you want to call it that. Similarly, for Macquarie Island, there's also been a couple of reports of the movement patterns of underyearlings, naive pups, if you want to call them that. With one of the first papers being an early 2000s paper by Bernie McConnell and colleagues, where they also showed this typical sort of southeasterly movement by the majority of animals, with one or two animals going sort of southwesterly from the island. This is sort of continued in subsequent papers, and you can see that there's a relative consistency between individuals in terms of the areas that they're utilizing. Interestingly, between these sorts of strategies where the animals go effectively east with the current, we'll get to that in a second, or west against the current, seems to potentially have further implications in their overall success. And there's a recent preprint that's been made available that seems to show just that, where animals that seemingly follow one strategy are more successful survivors than animals that follow the different strategies. So there's some potential selective pressure in terms of which direction animals actually go when they forage during their first year of life. So on Marion Island, it's important to understand a little bit about the background, the underlying environmental variables that potentially influence the movement strategies of animals from this population. So Marion Island is one of two islands that together form the Prince Edward Islands. It's located just under this SWIR on this map. The SWIR stands for the Southwest Indian Ridge, which is the dominant underlying bathymetrical feature. That. So the Southwest Indian Ridge is basically these white dots that scatter all along and follow right through to the northwest. It's a big divergent tectonic plate, which means that there are a few islands, these two islands to be exact, that are volcanic in origin, which are associated with these plates that are slowly moving apart. The nearest landfall to Marion Island are the Crozet Islands to the east, and these are about 1,000 kilometers east from the Prince Edward Islands. So Marion Island is the larger of the two islands, which is where we have the intensive mark resighting program of the elephant seals, and Prince Edward Island is designated as a high-level special nature reserve, which limits access to researchers, and we have a lot less information about what the elephant seals are actually doing on Prince Edward Island. What you'll notice here on this map is these sea level anomalies, and what you'll notice is there's some activity here associated with this area called the Andrew Bain fracture zone, and this is largely associated with the interaction of the dominant Antarctic circumpolar current, which is basically flowing very strongly in an easterly direction and interacting with the underlying bathymetry here to generate number of sort of mesoscale features. So this area is known to have a lot of eddy activity, for example, which is potentially exploited by a number of marine predators, and including to a certain extent potentially some of the elephant seals. However, even with these sort of bathymetric features, even though they are quite prominent, they're still very deep. So even the most shallow parts of the ridge are around 2,000 meters deep, with the rest of the sort of area being typically around 6,000 meters deep. So it's not necessarily that these sort of present sea mounts, if you want to call them that aren't easily accessible to many of the marine predators. Okay, the island itself is, as I mentioned, it's volcanic in its origin. So, which means that it basically rises very steeply from a surrounding ocean mass, and you sort of get a typical sort of scenery inland, at least, that looks like this, where it's just very hilly, with lots of lava flows, make traversing a little bit more tricky in places. But it's because of its size and because of its location, it's very oceanic in climate. So it's basically what that translates into is it's basically windy all the time and raining all the time, and it's a cool climate if you like to call it that. But the island has a number of accessible beaches that provide all-out spaces for many seabirds and seals. So penguins, there are 4 species of penguin that make use of the island. There are a number of other seabirds, albatrosses, petrels, cranes, and the like that nest on the island. And then there are the 3 seal species. So the 3 seal species being the southern elephant seals, which form these sort of rookeries in amongst the penguins, and then 2 species of fur seals, so Antarctic and subantarctic fur seals, both also breed on the island. So we've done a number of tracking studies already on older cohorts of Marion Island southern elephant seals, particularly focused on adult females, with a slightly smaller sample of adult males having been tracked, as well as then what we would term sub-adult animals. So these are animals that have, a sub-adult would be an animal that's a non-breeder yet. So for a male, that would typically be an animal that is younger than about 6 years of age. For a female, she would be under 3, sometimes 4 years of age, but an animal that has already survived its first year of life. So it's not a naive animal. These are animals that have some sort of experience, have survived sort of at least their initial foraging trips. And we've tracked in total about just under 100 animals. So I think there's about, what you see in this little animation is a total of 94 animals that have been tracked over the years, that have completed cumulatively about 150 odd foraging trips. So keep in mind, every individual animal would typically do sort of 2 foraging trips during the course of a year. One would be labeled typically as a post-molt foraging trip, and then the second one would be a post-breeding foraging trip. The post-breeding ones being much shorter, and being basically that short period of time between the breeding season and the molt, which sort of follows about 1 or 2 months later. And post-molt foraging trip being the longer foraging migration. So what you can see from this is that there's an overall trend in terms of the adult and sub-adult movement patterns to sort of exploit at least this sort of west-southwesterly direction away from the island. There are many individuals that are doing something different, that are heading east completely, some heading even to the African continent, and some heading down towards the ice itself. But the majority, there's a sort of preponderance of movement in a general southwesterly direction. And that sort of shows up in these wind plots that I've shown over here. What I'm showing here is this one on the top left are sub-adult females, and the bottom one are adult females. And what you can see for the females, at least, is there's a relatively strong sense of consistency between individuals in terms of their overall foraging direction. The same doesn't hold for males. Males are doing slightly different things, with some individuals going north. But at least for sub-adult males, maybe there's a trend that's similar towards that what we see for females. Now, the other thing that's important with these foraging migrations is that the elephant seals from Marion Island and also from other populations seemingly show a lot of fidelity to specific foraging strategies. So individuals that we've been able to track over multiple years and multiple migrations have shown a very high level of consistency in terms of the areas that they target. So what I'm showing you here are just two individuals, two adult females that we've tracked over multiple seasons. And these different colors show you just different years in which they were tracked. This is one individual over here. This is white 061. And each color then represents a different year. And you can see the lines don't necessarily lie right on top of one another, but they're pretty similar in terms of overall direction that the animals traveled in. Now, importantly, the timing of these migrations in terms of day of year and when they actually depart from the island and return to the island and where they are in the ocean during any given sort of time within a foraging trip also seems to be quite consistent. So that's what these little animations are showing you. This is just basically the location within a track plotted over the day of year. And what you can see is, as far as day of year goes, the animals sort of all, the clusters seem to be pretty close together as the animal proceeds. Similarly, for the animal in the top right-hand corner here, this is yellow 189, and she's one of the animals for which we have the highest number of tracks. And you can see these clusters are all sort of quite close together, with this little yellow greenish dot joining a little bit later. That was her very first foraging migration that we tracked there as a sub-adult female before she was breeding. So that's why it's sort of a little bit of an outlier compared to the others. But I mean, in terms of the actual dates that she departs from the island and returns to the island, those normally the dates that she actually hauls out back for after a molt, after a post-molt foraging migration, are normally within 1 day of one another. So we can check the date that she returned to the island. Normally is the next year she'll be back either on the same day or at the most 1 day earlier or 1 day later. So there's this high level of fidelity in terms of once an animal seems to have a strategy that works, where they've survived it, that they would then return to that strategy and keep doing that year after year. And this seems to hold basically across the board, both for males and females. So we were interested, of course, in what the pups were doing on these maiden foraging trips, where they have not had the opportunity to gain any experience, where they've not had the opportunity to learn from the conspecifics. And we wanted to see, well, how does this then develop? What sort of strategies are these pups employing? We instrumented a total of 79 recently weaned pups with GLs, geolocation devices. And these tags were then deployed, as you can see there, between the years of 2005 through to 2008. We instrumented a total of 44 male pups and 35 female pups. Importantly, most of these geolocation tags were instrumented right after weaning, so before the maiden foraging trip. But some of them were also instrumented still during that first year of life, but after the maiden trip. So when the animal returned for that sort of winter haul-out period, when they still come back to the island just to rest, seemingly, some of those were instrumented then as well. So for some animals, we have the maiden foraging trip, and for others, we have sometimes the second or the third foraging trip. But all of those were still within the first year of life before the first molt. And then for some of the animals, we were able to get tracks that were actually covering multiple trips. So we have 1, 2, and 3, for example. So what were we expecting to find when we started looking at the results from this? Well, firstly, in terms of track distances, we would have expected some sort of gradual increase in the foraging track distances that animals would venture from Marion Island, as well as the total track distances. So in terms of whether an animal sort of gets to a point that's really far away from the island, we would sort of expect them to potentially extend that sort of time period that they're away from the island and correspondingly increase their total track distances over time. Why I'm showing you the plot on this graph is this is the type of findings that have been found for, or reported at least for harbor seals. And what you can see here, these are also subsequent or multiple foraging trips or initial foraging trips by harbor seal pups. And you can see the overall distances that they swam sort of gradually increase with age as they age over here. Apologies if this is a bit small. You've got age on the x-axis over here, so it's 0 up to 400 days. And then this is distance that was swum per day. And you can see that animals were increasing their distances that they were traveling over time. So this is sort of one of the expectations we had. The second expectation that we had was potentially comparable travel directions to what we've seen with older cohorts from the same population. This is largely associated with the idea of if there's some sort of innate response to environmental variables or environmental cues, then surely the overall travel directions and approaches would be similar to that of other animals that we've tracked before. And this does seem to be, at least for the reports for some populations, potentially the case, at least from just the basic sort of cursory look at what the animals were doing. So for example here, for the King George animals that I mentioned earlier, what you see on the left-hand plot here, these are those same under-yearlings, the weaned pups. And on the right-hand side are adult females from the same population. And what you can see here, or what you'll notice immediately, is that the travel directions are similar. So the adult females also concentrated most of their foraging sort of towards the west of the Antarctic Peninsula. But the key difference here being that the pups were not sort of entering the higher ice density areas as efficiently, at least, as the females were. But the overall sort of travel directions were similar. So this was sort of one of the expectations we had from the pups that we were instrumented from Marion Island. Another expectation we had was that there'd be some sort of consistency in travel direction during initial foraging trips and between those initial foraging trips within the first year of life. So as I mentioned, the adults seemingly show a very high level of consistency and fidelity to foraging areas. So we therefore expected that once an animal survives a foraging trip, that it would likely return to the same area in a subsequent foraging trip in order to, and then sort of stick with that sort of strategy. What did we find? So firstly, it's important to note that from those 79 instrumented animals, we were only able to recover 34 geolocation tags. This is a result of tags either being lost at sea, so some of the animals returned to the island without their tags, while a number of the animals potentially didn't survive their first foraging trip. So there were quite a few animals who were never resighted again during the resighting efforts on the island, so were presumed to have not survived their first foraging trips at sea. And unfortunately for those, we don't know where they went because the location tags are archival in nature. So we have no way of remotely monitoring them until we get the instruments back. From the 34 tags that we were able to recover, unfortunately, some of them were corrupted. The data files on them were corrupted, and we were able to obtain useful light data in order to calculate their locations from only 16 of these tags. But fortunately, those 16 tags included a total of 29 foraging trips. So for most of the animals that we recovered useful tags from, we got multiple foraging trips in that sort of first year of life for them. Now, what you see here, this little animation that's been running all along are those locations that we have for the tracks. And what you'll immediately notice here is that it doesn't seem to be a particularly clear pattern. Animals are heading in all kinds of directions. There's potentially a general east-west axis, if you want to split it that way, and perhaps less of a north-south sort of general direction of travel. But individuals are not doing the same thing, and between individuals also not. We'll get to that in a second. What you'll notice here is there's one outlier here. This animal over here is Pink 116. He was a pup. He was born in 2006. and instrumented then. And in fact, this animal traveled the furthest of any of the animals that we've instrumented, including the adults. This animal covered a total distance of around 16,000 kilometers during which would have been his first foraging trip. Successful return to the island, but something that's completely different to what any of the other animals are doing. So when we started analyzing some of these results, we noticed a couple of things. First thing is that between these trips during the first year of life, there was a decrease in their overall duration. So what that means is when an animal, the first maiden foraging trip, tended to be a couple of days longer than the second one, which in turn was a couple of days longer than the third one. So on average, the trip durations, the first trips that animals undertook were around 140 odd days, and the second ones were closer to 90 days, and the third one's around about 74 days. This was, so what you see here is on this plot is just a prediction from a general linear mixed effects model, in which we illustrate this sort of trend in trip duration and how it changes or how it decreased between foraging trips within that sort of first year of life. Now, the overall durations of these trips were, the first foraging trips were tended to be longer than the average post-breeding foraging migrations of adults, but shorter than the post-molt foraging migrations of adults. So that was sort of maybe within something that we would have expected. Now, when we started to have a look at the actual distances that animals covered, we started to see some interesting patterns, but we found no evidence for any real effect associated with the trip number, with the sex of the animal, or the year in which the animal was instrumented in. The reason why we're interested in the year that an animal was instrumented in was potentially larger scale dip variation in terms of food resource availability, and things like that would potentially influence we would have expected, would have influenced the foraging track sort of characteristics within individual years. But so what you see on this plot over here, what I'm illustrating here is the black lines and dots are the maximum distances that animals reached from Marion Island during these foraging trips, and then the blue dots are the total distances traveled. So if you accumulated all the travel that we have recorded for an animal, what is the total distance that an animal traveled? And this is plotted over the trip duration. And what you'll notice here, this is trip 1, trip 2, and trip 3. And what you'll immediately notice here is that there seems to be a little bit of a trend in terms of the total distances that animals traveled. There's maybe a slight negative trend. This is not statistically significant. There's no, but if you just sort of look at it visually, that seems to be roughly the case. There's one or two individuals like this one over here that did some extreme distances during that second foraging trip of theirs. But interestingly, the difference between the maximum distance that animals reached from Marion Island did not vary at all. There's hardly any sort of suggestion here if you compare between these foraging trips. This sort of line, it's got a relatively similar slope, and the actual distances, the range of distances, all overlap completely between the foraging trips. So this may be, I'm saying maybe in a very sort of questionable way, may be suggestive of potentially some more directed movements in terms of as an animal progresses. So what that means is during the first foraging trip, even though an animal didn't go that far away from the island, it still covered a very long distance. So perhaps performing more sort of convoluted tracks and more sinuous movements, if you want to call it that. Whereas in subsequent foraging trips, it started to direct its movements more, reaching its destination perhaps in a more sort of direct fashion rather than a meandering way. Now, when it comes to the actual sort of trip directions, we see something that looks a little bit like this. So maybe just to quickly explain the plot, these are again just the same type of wind plots as I showed you for the adults and sub-adults. But it's split here by categories. So firstly by sex, with females and males, and then trip number between trip number 1, 2, and 3, and then the year of instrumentation again. Now, what you'll immediately notice is that there's as you would have seen from the little animation, that there's not a very clear trend. This trend here, this sort of southwesterly trend, seems to be something that's maybe worth looking at. But what sort of throws one a little bit potentially is this red line, which is associated with that one animal that traveled at extreme distance, which sort of skews this plot a little bit. And that's that one, that pink animal that traveled the 16,000 kilometers and reached the greatest distance from Marion Island. But we found no evidence for any effect of trip, so no differences between trip numbers in terms of travel directions, no differences between sex or between year. What we used was a circular mixed effects model, and basically what this model did was it showed an improvement in fit when we included these variables, when we included trip, sex, and year, but there was, with the posterior estimates, this is the Bayesian model, with posterior estimates, there was substantial overlap between these different models with the different variables, suggesting that there's no significant or no sort of standout effect associated with each of these variables. Now, furthermore, when we start comparing the sort of overall tracks between or within individuals, so between foraging trips that were covered by single individuals, we also see no real evidence for any consistency between foraging trips. So, for example, what you see here, this is animal pink 003. These little orange lines, excuse me, is trip 1, and this is trip 2, the blue dots, and they're in completely different directions. So there's no, seemingly, the animal was doing one thing, trip 1, came back, rested for a bit, and then headed off in a completely different direction on trip 2. It's a little bit more convoluted for other animals, but there's no real overlap in terms of individual trips, in terms of the areas that animals were using. So there's no suggestion that there's a lot of consistency between their foraging trips. So if we revisit our sort of expectations, we sort of start thinking about each of these sort of these outcomes, and we sort of have to start, my slide is doing something funny here. We have to start sort of thinking about each individual outcome. Now, the first one, where we expected this sort of gradual increase in foraging track distances that animals ventured from Marion Island in total track distances, we found no support for in our results. Similarly, when it comes to the expectation of comparable travel directions, when we compare them with older cohorts from the same population, we found no real support for that. And then lastly, we found no support for consistency in travel direction, at least not during those initial foraging trips. So, and these sorts of expectations then speak to those variables that we were talking about right at the start, associated with consistent reaction or behavioral cues associated with environmental parameters and so forth. But then rather, what we're thinking is happening is that animals are likely learning rather more through experience. So there are a couple of other things that we did not extensively consider that may help to explain some of this in our results. And one of them is the potential that animals are responding to sub-mesoscale environmental cues. What I mean by sub-mesoscale are these sort of smaller filaments of water movement that potentially concentrate prey in smaller scale than what we are actually considering with our assessments to date. So we know that mesoscale features, we know that eddies and meanders are important for many of the top predators, including elephant seals, and that some of the older, or at least the larger animals are using them and potentially responding in a predictable way to some of those features. But we haven't been able to, at least not with the pups, consider the smaller, finer scale environment. It's important to note that with the geolocation devices we've been using, the accuracy, the actual accuracy that we can get the location estimates down to is at a scale that's not suited to these sorts of investigations. So we sort of are in need of finer scale location estimates and more accurate location estimates if we want to start sort of assessing the potential influences or the potential of individuals responding in a predictable way to environmental cues, or at least these fine-scale environmental cues. Broad-scale environmental cues, such as the overall transport of Antarctic circumpolar currents and mesoscale features, seems to not be anything that we've picked up, at least in our analyses. Another potential explanation for these sort of trends that we're seeing is that there may just be simply multiple behavioral phenotypes within the population. And this sort of leads to the question of whether this is maybe a form of some sort of within-generation bet hedging strategy. So the rationale behind this is that if you have a really unpredictable environment, particularly an environment that is temporally unpredictable, so differs between individual years, with some areas being really productive and safe for animals to access, and some areas being really dangerous, but sort of shifting in between individual years, then at a population scale, it may be beneficial to have multiple phenotypes, where some animals would head off in one direction, some animals would head off into a different direction. And if they survive, then at least some, or if they survive, they survive. If they don't, they don't. But at least you'll have some that get to an area that is that was sufficiently profitable for them to survive the first year of life. This is perhaps a little bit of a fanciful hypothesis, if you like. There's a lot that needs to be sort of unpacked, and we don't have the information yet, but I think it's a hypothesis that's worth exploring in future studies. Of course, what this calls for and what we continue needing is more long-term, longitudinal studies, whereby we have individual animals that we know their breeding histories, where we know how successful, what their fitness is in terms of producing offspring are, and where they are between individual years and how that varies between individual years. So with that, I'd like to just thank you all for your attention to the presentation. And of course, with this work, there's been a lot of funding bodies involved. So I'm just highlighting some of the organizations on this last slide. And I'd also just like to point you out to the Marion Island Marine Mammal Program website. If you're interested in the overall work that's happening on the island in terms of work with seals, killer whales, please have a look at the website and give it a thumbs up. Thank you for your attention.
Well, Trevor, thank you for a very interesting presentation. It's incredible the amount of effort and work that's gone into this. And one of the things that really strikes me about it, and I had similar experience over my time working on pinnipeds with instrumentation, looking at foraging, is if I recall, you started out with 79 animals instrumented, but in the end, your sort of workable sample size was about 30% of that. And it leads me to ask a question quickly of your lack of finding effects. Do you have a sense of what your power to detect differences of those possible effects are? I'm guessing it probably wasn't as strong as you might ideally like.
Yeah, sure. It's so there are a number of limitations in terms of how far we can take this, particularly with that sort of sex split, with only a very small number of female pups who we were able to track compared to a higher number of male tracks. And this sort of speaks into the larger population trends also, where females are the ones that really show this very high fidelity to foraging strategies, and consistency between individuals in terms of what they're doing. Whereas males are sort of, even though they show high fidelity to strategies, there's big variation between individuals. And that may be something that sort of befuddled a little bit our outputs here in terms of which is associated with the sample size. So absolutely, it's one of those. It's difficult to overcome problem, as you well know, and as you mentioned, the effort that goes into getting these tracks is very high. And look, I mean, at the same time, so these were some relatively early days sort of geolocation tags. Tags are improving all the time. Attachment methods, people are getting better at doing it, and with potentially with enough support, one could improve on this going forward.
Yeah. I don't know if it's been a while since I've done any kind of tag deployment. I don't know if they're also getting cheaper to get large numbers, but that was always a factor in how many deployments you could make. Forget the logistics of getting them deployed. I don't know if we have any other questions yet that's come along. If not, let me ask you one other question that if it's something you've looked at, and that is to what extent there might be variability in development of diving capability, and whether that there might be some sex effect in that, or effect of individual weaning size or something like that. Have you looked at that in any way?
There's been a little bit of work, but not with the weaned pups, not with the under yearlings. So the tags that we've deployed on them to date have all just been location only devices. But there has been a little bit of work. There's a paper by a student actually that came out last year, Kiara Lush, from the University of Pretoria, that looked at the ontogeny of some of the dive behaviors, but looking at that sort of older cohorts. And those are seemingly reasonably predictable, where you've got this sort of increase in dive depths and durations as animals mature. But it's so there's been a little bit of work, but we're sort of missing that crucial first couple of foraging trips.
Okay, thank you much. I guess we do have at least one question that's popped up, so I'll leave it to that.
Yes. There's a question, as the weaners are heading off in various directions for different trips, any thoughts on how they are getting back successfully?
No, I'm hesitant to I mean, the actual navigation capabilities of seals in general. There's been a lot of sort of circumstantial and ideas and hypotheses thrown out, whether it be associated with water movement and current movement, or whether it be associated with geomagnetism. I'm hesitant to venture against that. I simply don't know in terms of elephant seals how they actually are able to navigate this way. I mean, and the same goes for adults. I mean, there probably are landmarks to a certain extent from potentially from a bathymetry perspective, but most of those landmarks are really deep. We know that elephant seals can and do reach really deep depths, but how would they sense anything at a depth of 2,000-3,000 meters from a landform perspective? I sort of doubt. So it's probably something to do with current movements that they're able to navigate broadly, but that sort of finer scale navigation, I don't know.
That's all right. Next question is is there a difference of food preferences of different animals depending on foraging trips?
That we don't know either. So from a diet perspective, there's been quite a bit of work done, and we know for the pups, we know that they take quite a substantial amount of krill while they're foraging, and we know for adults that seems to switch then to rather a diet more on McTurford fish, and so yeah, so there's this sort of there's this shift as age that's associated with specific foraging trips. We don't have that sort of, there is a bit of work. We have, there's a colleague of ours, Dr. Nico Lupker, who's actually doing a postdoc in the US at the moment, he's an isotope, stable isotope specialist. And he has been working a lot on the stable isotope samples from the Marion Island elephant seals. We were in discussion a while ago about trying to tie in some individual foraging tracks with the diet information we have for those individuals, but that hasn't been done yet. So I simply don't have an answer yet, I'm afraid.
Great. And I had a question too. You talk a lot about the foraging preferences of the pups, but considering they are pups, is there any thoughts given to potentially predator avoidance behaviors at those young ages?
Sure. Sure. Yeah, I think that probably should that goes hand in hand, and I think which one outweighs the other, I don't know. But I would imagine certainly predation levels during that first year of life are really high. We know that many of the pups are taken immediately adjacent or around the island. There's been some work quantifying that. We know killer whales are abundant, and they take out quite a number of pups as they leave. Further from that, we don't know causes of mortality, but it most likely would be some sort of combination of starvation and or predation. Which one, once they sort of, in my mind, I sort of view it as there's a sort of danger zone immediately around the island where you've got predators circling all the time. But once they sort of head past that, now having to find food, two risks, whether that is finding enough food or avoiding predators. I don't know which one will carry sort of the biggest threat to survival during the first year. Probably both.
Yes. And there's one more question that has anyone started to look at any correlation between oceanographic change anomalies in association with the variation in foraging patterns?
There was some early work, but it wasn't very definitive. So there was a couple of years ago, we've had one or two students who've had a look at some of the variation in terms of particularly eddy activity, and if that differs between years and what individual elephant seals were doing between years. But there wasn't a very conclusive sort of outcome from any of it. So it sort of has been done, but there's probably scope for a lot more. I think there's been a little bit more work done, particularly on other populations. So the Kerguelen population, there's been quite a few papers that have shown these sort of associations with oceanographic features and how foraging strategies seem to be influenced by them.
Thank you. That's all the questions from the audience that I can see currently. Marion Island looks super fun. How can students join the Marion Island program if they wanted to be interested in that?
So the best thing to do, well, what I would advise is have a look at the website. And then, so the current principal investigator on the project is Professor Nico de Bruin at the University of Pretoria. It would probably be a good point of contact. His details would be on the website, and they can get in touch with him, and he can probably direct them a little bit. Admittedly, The overall program is run through the entire South African system, which makes it a little bit more tricky for international students and scientists to get involved. It's not impossible. We have had particularly international postdocs involved in the program. Undergraduate students, it's a little bit more difficult logistics-wise. It's very difficult to get people onto the island because we're reliant on the South African National Antarctic Program and their ship and all their logistics. So there's it's perhaps not that easy, but I think Nico is a sort of a good first point of contact, and he would be able to sort of advise and give some pointers to anyone interested in getting involved.
Excellent, thank you.
So there was one more question that popped up if you want to follow up on that, and then I think we'll call it, since we're a little after two my time, but past the hour. So.
Yep, there is a question here. Has any of the tracked weaned pups had their mum, dad, or dad tracked at one point? And has any of the tracked weaned tracks later? Has any of the tracked weaned tracked later on in its life? So I guess two questions here. Did we track any of the parents at the same time as the pups, or were the pups tracked through multiple years?
So we don't have parents who were tracked at the same time as pups. We do have there are some tracks for pups once they're a little bit older, where we have their tracks, and also yeah, I think we do have, but they're very few. But what we do have is some of these weaned pups. We do have tracks of the same animals as adults. So, for example, that pink 003 animal that I showed you with the two tracks that separated out in two complete different directions, he grew up to become a beach master on the island, and we have a couple of tracks for him as an adult as well. So that's something that when I initially started looking at these tracks, that was one of the sort of first questions. But the sample size is it gets smaller and smaller as you go, as you start adding something to it, and becomes really difficult to make any real conclusions from there. But we have a handful, yes.
I think it raises a really interesting point here, is that asking the really interesting questions makes this kind of work one that has to be a lifelong process, of which the payoff comes very late in your career. So
yeah, that's a good point, I think. Yeah. True.
Yeah. Well, with that, I thank you very much for interesting and exciting presentation, and I appreciate your willingness to do this for the society. So thank you very much, Trevor. Okay, with that, I guess we'll say goodbye to everyone, and tune in for a seminar next month. Thank you very much for everyone who attended. Bye-bye.